Hello everyone. Today we will discuss the metabolic functions of liver and its supply. So how does the liver works? It metabolizes the product which is coming from a GIT. So before knowing it, we have to understand the physiological anatomy of the liver. So the liver is the largest internal organ of the body and its basic functional unit is liver lobule, which we also call sometimes hepatic lobule. And where it is present in our body, it is present in right upper quadrant. Now how this liver lobule looks like? It looks like a cylindrical structure 0.8 to 2 mm in diameter, and about 50,000 to 1 lakh liver lobule are present in a liver. Alright, so this is hepatic circulation. So the liver is supplied by the hepatic artery as well as the portal vein. So this blue thick vein is portal vein, and how it is formed? It is formed by the SMV is superior mesenteric vein, and this thin blue vein which is coming from the spleen is splenic vein. So when the splenic vein is combined with a superior mesenteric vein, it forms the portal vein. About 75% of the blood is supplied by this portal vein. Now this portal vein is again split into a venules, and then these venules, they are converted into sinusoids. And what are sinusoids? Sinusoids, these are low pressure vascular channels. And then these sinusoids, they drain the blood from the central vein. They collect the blood and then drain via hepatic vein into a IVC is inferior vena cava. Now the liver have other supply also, which is the hepatic artery. This is the hepatic artery, which is the branch of aorta. All right. Then this aorta, this hepatic artery is split into a arterioles, and this again supplies to a hepatic lobule. In this way, the liver is supplied by the hepatic artery as well as the portal vein. And in this with the portal vein, it collects the blood from the GIT, and now the blood enters into the liver. Now, this is the hepatic lobule or liver lobule, which was the basic functional unit of the liver. So this liver lobule, it is constructed around the central vein. This is the central vein. These are the sinusoids. Now, this portal vein, red hepatic artery, and a green bile duct, these together call a bile duct. So each hepatic lobule at the corner, there is presence of this portal triad. And these orange color cells, what are these? Any guesses? Yes, these are hepatocytes. And this green canaliculi is a bile canaliculi, which collects the bile, and then it comes ultimately into the bile duct. This white portion is called spaces of Deci. This spaces of Deci have a very importance here because it helps in exchange of the products between the cells as well as between the blood. And these spaces of Deci, when this extra fluid is present, they drains the extra fluid into lymphatics, and then from lymphatic into the lymphatic duct. In this way, they help in exchange. Now, there are other cells also. We can see here this cell. These are not hepatocytes. So, what are these? Yes, there's mention here. These are endothelial cells. These endothelial cells have a fenestrations. So, it help in exchange of, or the large protein molecule can cross over from it. And then the Kupffer cells. Yes, these Kupffer cells, they help to phagocytose the bacteria because the blood is coming from the GIT. So it might contain a large amount of bacteria there. So this is all about the hepatic lobule. This is the liver. We have taken out a small section of the liver, then we zoomed it, and this is the hepatic lobule with a central vein. Again, a zoomed portion of this one is hepatic lobule. So what are the functions of liver? So the liver is a reactant pool. So it performs a lot of function. But the main functions are metabolic function and secretion of bile, which we will discuss in coming slides in detail. The third function is a storage function. Liver stores the fat-soluble vitamin like vitamin A, D, E, K, and also iron in the form of ferritin. It plays role in blood coagulation as it forms the clotting factors. It can be also work as a reservoir of blood because as liver cells, the hepatocytes, they are expandable. So when the body needed extra blood, it can store it about 1.5 liter more blood in them. As it have a cover cells, so it helps in phagocytosing the bacteria and it clears the blood. Catabolic and excretory function of liver. Liver excretes drugs and hormones like thyroxine and steroid, and all the products of the reactions excreted into a bile and then return into a bloodstream and ultimately excreted via kidney. It excrete and catabolize by two metabolic reactions. The phase 1 reaction is oxidation and hydroxylation by the help of cytochrome P450, and the phase 2 reactions are conjugation reaction with glucuronic acid and sulfate. Most important function, metabolic function, and the carbohydrate metabolism. So if the blood have more glucose, then this liver is going to form a glycogen because it have to store the glucose in the form of glycogen. So it stores the glycogen, and the process of conversion of glucose into glycogen is we called it glycogenesis. And when a lot of glucose and so it is occurring glycogenesis. But when in the blood, because the liver is doing its buffering, glucose buffer function, it's a job of liver. So when there is less glucose in blood, then it convert the galactose and fructose into a glucose. And how it is going to produce extra amount of glucose? By gluconeogenesis, which is a synthesis of new glucose from one carbohydrate source like amino acids, pyruvates, and fumarates. And there are other cycles like glycolysis and glycogenolysis also occurring here. And what is the stimulating factor for glycogenesis? Yes, it's the insulin, which is secreted from the pancreas in a well-fed state. This is the two pathways occurring side by side. The green arrow is for the glycolysis and red is for the gluconeogenesis. Here we can see that formation of glucose from a non-carbohydrate, so lactate, glutamate, and pyruvate ultimately form the glucose. Second metabolism is fat metabolism. The fat is first split into a fatty acids and glycerol, and then there is the oxidation of fatty acids to supply the energy for the body function. Liver also plays role in synthesis of cholesterol, phospholipids, and lipoproteins. as well as it synthesizes the fat from the protein and the carbohydrates. Triglycerides, phospholipids. Phospholipids, they can synthesize the fatty acids. Then these fatty acids undergo beta oxidation, formation of acetyl-CoA, TCA cycle, and generation of NADH and FADH2. And then it enters the electron transport chain and produces a lot of energy. Okay, so this is the carbon dioxide which react with the ammonia and form carboyl phosphate synthase. This is carboyl phosphate and which react with ornithine and form citrulline. This citrulline enters the cytosol and it converted into a arginosuccinate. Then it is split into a fumarate and arginine, and in presence of arginates is formed ornithine and urea. Then in this way the urea is excreted out of the body. Now the second most important function of liver is secretion of bile. About 600 to 1000 ml per day bile is secreted. And what it is a micellar solution in which major solutes are bile acids, phosphatidylcholine, cholesterol in the ratio of 10:3:1. And when this ratio alters, then this formation of stones, which we call bile stones. The function of bile is emulsification of the fat and digestion of the fat. This is the biliary system, which is formed by two ducts coming from the liver, then which combine with the cystic duct, form common bile duct, then common bile duct, the bile comes, and then it enters the duodenum when this relaxation of sphincter of Ori. This is the liver bile and gallbladder bile, which is quite different because there's a concentration of bile occurs in the gallbladder bile. And it consists of water, bile salt, bilirubin, cholesterol, fatty acids, lecithin, a lot of ions. And why it contains these? Because it has to emulsify the fat. This is the transport process of cholangiocyte that secrete alkaline-rich fluid and reclaim useful product. With these aquaproteins, they add more and more water in the lumen, and because this is the which side? This is the apical side, which is towards the lumen, and this is the basolateral side, which is towards the blood or extracellular fluid. And in this way, it adds more bicarbonates and the water in the lumen. This is the concentration of bile while storage in the gallbladder. So water is getting inside and the bile is concentrated. Sodium also coming inside, hydrogen is going outside, as well as chloride is also coming inside via these channels and pump. This is the bilirubin conjugation. Now, the cholelithiasis is the formation of stones. So the gallstones, they can be two types. They can be pure, they can be mixed. So pure gallstones, they can be cholesterol stone, pigment stone, calcium carbonate stone. And the mixed stone, they can be 50% cholesterol, calcium salts, and bile pigment. Because they mix, they contain all of these things. Okay, here we can see a big solitary white stone. So this is a cholesterol stone, which is single and white, and the surface is very smooth. The size is approximately 3 centimeter. So these are pigment stones. They are black. They are multiple, and then the calcium carbonate. So the calcium carbonate, it contains the calcium carbonate these stones, and they are also multiple, and they are chalky white in color. Then these are the mixed gallstone, which are multiple and they are multifaceted. Like they have different. See this one, multifaceted. This is the formation, pathogenesis of the gallstone. How it is formed. So if there's any alteration in the mixed micelle, then this formation of the gallstone and production of lithogenic bile. If there is alteration in these enzymes, more HMG-CoA, more cholesterol, less 7-alpha OAs, there is less bile acids, decrease multi-drug resistance receptors, there is decreased lecithin. So in this way, it's altering the normal mixed micelle. And then there is supersaturation of the bile, cholesterol nucleation, gallbladder hypomotility, and biliary sludge as well as formation of the stone. Thank you.